Hello everyone, this is Minoy Cristobal. I am a doctorate student in Dr. Hansenstein Monogastric Nutrition Laboratory. Today I will be discussing one of our latest research titled The Effect of Feeding Intact Protein from Soybean Meal Instead of Crystalline Amino Acids on Energy and Nitrogen Balance by Growing Pigs. This research was presented at the ASAS Midwest meeting in March of 2024. From almost all soybean meal produced in the US, near 80% is used to feed broiler chickens, pigs, and laying hens, and the rest is used to feed other species. This is because soybean meal is the main protein ingredient in diets for monogastric animals due to its adequate balance of indispensable amino acids. Another way of supplying indispensable amino acids to diets is through the inclusion of synthetic amino acids. This is an industry that has experienced a steady growth through the years. For example, in 1950, with methionine becoming commercially available, years later with the production of lysine, followed by threonine and tryptophan. Nowadays, it is possible to find all indispensable amino acids in its synthetic form, and these are mainly produced by three methods. With the inclusion of synthetic amino acids, it is possible to reduce the inclusion of protein ingredients such as soybean meal and therefore have diets with reduced crude protein. The effects of reducing crude protein in diets for pigs has been addressed by several studies. In general, the studies reported a lower nitrogen excretion, no negative effects on growth performance, increased net energy of diet, and reduced incidence of post-weaning diarrhea and lower feed costs. However, some studies also reported a negative effect on nitrogen retention, more fat and less muscle deposition, and changes in intestinal morphology of pigs. Now, for this study, we focused on the effects on nitrogen balance and metabolism of energy. Therefore, our objective was to test the null hypothesis that reducing crude protein in diets by reducing soybean meal inclusion while adding synthetic amino acids in diets fed to growing pigs, 1. Increases nitrogen retention, and 2. Increases metabolizable energy. Moving on to materials and methods, we formulated 4 diets. Diet 1 contains 60% corn and 35% soybean meal, some soybean oil, and no synthetic amino acids. This diet contained 20% crude protein. Diet 2 was similar except that we included 3 synthetic amino acids, and with that, soybean meal inclusion was reduced as much as possible but to still meet the requirements of digestible indispensable amino acids for growing pigs. This diet had a total crude protein of 16%. For the next diet, we followed the same logic but with the inclusion of 4 synthetic amino acids, and crude protein of the diet was reduced to 15%. The last diet included 5 synthetic amino acids with a total crude protein of 13%. In the last 3 diets, the synthetic amino acids were added in the order of most limiting amino acid for growing pigs, and as we reduced soybean meal inclusion, we increased corn inclusion. Now, let's take a look at the analyzed composition of diets. The 4 diets are represented by their formulated crude protein content and with different colors. We can observe that the analyzed crude protein in diets were close to the formulated crude protein, indicating that the diets were mixed properly. The acid hydrolyzed ether extract values were below 5% and not much difference among diets because corn and soybean meal have little fat content, and that is between 1 to 3%. And all diets had the same soybean oil inclusion. And there was no much variation for ash content among diets. Looking at the analyzed total amino acid concentration in diets, we observed that in general the values were close among diets, and maybe the concentration of some amino acids in the diet with 20% crude protein is greater compared with the other diets, and this is because that diet didn't contain any synthetic amino acids, and therefore it may have more amino acids in excess. Now, for animals and experimental design, pigs were placed individually in metabolism crates. Experimental diets were fed for 5 days of adaptation to the diets, followed by 4 days of collection. Feces and urine were quantitatively collected, and fecal samples were oven-dried and analyzed for dry matter, nitrogen, and gross energy, whereas urine samples were freeze-dried and analyzed for nitrogen and gross energy. A total of 40 barrels and gills, around 20 kilos, were used. Pigs were allotted in a randomized complete block design with 2 blocks of 20 pigs in each block. There were a total of 10 replicate pigs per diet. Data were analyzed using prog mix in SAS, and diet was the fixed effect, and replicate and block were the random effects. Contrast coefficients were used to determine linear and quadratic effects. Moving on to the results, for apparent total tract digestibility of dry matter, of diets, there was a quadratic reduction as crude protein in diets was reduced. Similarly, for apparent total tract digestibility of gross energy was linearly reduced as crude protein in diets was reduced. These results indicate that the digestibility of dry matter and gross energy in soybean meal might be greater than in corn. That's why, as we reduce soybean meal while increasing corn inclusion, the digestibility of dry matter and gross energy of diets is also reduced. For digestible energy in diets, we observe a quadratic reduction as crude protein in diets was reduced. This indicates that soybean meal contains more digestible energy than corn, and as soybean meal inclusion was reduced in diets, the digestibility of diets was also reduced. This agrees with NRC 2012 and digestible energy values reported for soybean meal and corn. However, for metabolizable energy, there was no effect of reducing crude protein in diets. The fact that the metabolizable energy did not increase as soybean meal was reduced while corn was increased indicates that metabolizable energy concentration in soybean meal must be higher and closer to the metabolizable energy in corn, and this differs from NRC 2012 that reported lower metabolizable energy concentration in soybean meal compared with corn. Nitrogen absorbed was calculated by the difference between nitrogen intake and nitrogen excreted in feces. There was a linear reduction in nitrogen absorbed as crude protein was reduced in diet. Nitrogen retained was calculated by subtracting nitrogen excreted in feces and nitrogen excreted in urine from nitrogen intake per day. There was a linear reduction in nitrogen retained as crude protein was reduced in diet. These results might be explained by the fact that as crude protein in diets was reduced, the nitrogen intake per day was also reduced. The upper and total tract digestibility of nitrogen was linearly reduced as crude protein in diets was reduced. This indicates that nitrogen from soybean meal is more digested than the nitrogen from corn. For nitrogen retention as a percentage of nitrogen intake, there was a linear increase as crude protein in diets was reduced. For nitrogen retention as a percentage of nitrogen absorbed, also referred as biological value, the values ranged from 77 to 90%, and these values were greater than previously reported. And we observe a linear increase as crude protein in diets was reduced. These results indicate that pigs utilize nitrogen more efficiently as crude protein in diets is reduced by reducing soybean meal while increasing corn and supplementing with synthetic amino acids, meaning that the amino acid concentration in diets with reduced crude protein might be closer to the amino acid requirements by growing pigs. Based on the results obtained in this study, we cannot reject the null hypothesis 1 and conclude that reducing crude protein in diets by reducing soybean meal inclusion while adding synthetic amino acids reduced apparent total tract digestibility of nitrogen but increased nitrogen retention by growing pigs. However, we reject the null hypothesis 2 that was that reducing crude protein in diets by reducing soybean meal inclusion while adding synthetic amino acid increases metabolizable energy in diets because, as we observed, there was no effect on metabolizable energy in diets fed to growing pigs. We also conclude that retention of nitrogen by growing pigs fed corn soybean meal diets is greater than previously reported. With that, we would like to thank the United Soybean Board for its support of this work and the team at the Smith-Buckling Corp for their helpful discussion and guidance. Thank you for your attention, and if you'd like to learn more about our research, please visit our website.